NK cells, 20 to 40% of our white blood cells is lymphocytes. Okay, so when you get the blood and you look for the leukocytes, white blood cells, 20 to 40% of them are sort of lymphocytes. And where they usually, you can see them, the main place for them, of course, they're in the circulatory, but their base for them is lymph nodes. And 99% of the lymph node is literally lymphoid cells we have. If they're inactive, if they have not encountered the antigen, we regard them, we say they're naive. And you can see the picture here. I don't know, does it show you good? Let's do cheating, if it helps. Yeah, you can see better now, yeah, full size. So this is a single lymphocyte. It's around 6 micrometer diameter, and the majority of it is its nucleus. Okay, so tiny cells compared to other immune cells, well, one of the smallest cells. Majority of it is nucleus, and out of these lymphocytes, they realized they came across some cells which do not express the classical lymphocyte marker. Instead, they express CD56. Then they realized their role also is in targeting tumor cells or targeting cells which have been infected with virus, and they have this direct killing mechanism. Therefore, they call them natural killer cells. The main marker for them is CD56. Okay, the other lymphocytes we mentioned earlier on, B lymphocytes. What was the marker for them? CD19. Yes. T helper cells, marker for them? CD. No. CD4. CD4 helper cells, do you remember? Cytotoxic, CD8. So they literally have a central role in activating the immune system. This one, cytotoxic effect, targeting cells which are virally infected or tumor cells. In this slide, okay, the big blobs you can see as white. Okay, this is an experiment I did myself. We had 2 different cells. The red cells are those immune cells. We call them they're T helper cells. Okay, which they also the other name for them once they're active is effector cells, T effectors. I labeled them as a red fluorescent. The green one are the cells which switch down the immune system. You remember the scale showed you immunity and regulation? These are immune regulation cells we have from the T cells, T cell subsets. They're called regulatory T cells. They regulate the T cells. We have a lecture on that, don't worry. But on this video, what you can see, I'll play for you. I put regulatory cells, switch down the immune system, and those which switch on the immune system, the T effector or T helper cells. And next to it, I put the dendritic cell as well. Now you can see what happens like this. The dendritic cell is migrating, getting connection to various different cells, you see? And also like this one here. Okay, like that one there, moving around. So in reality, in our system, this is just in vitro. We have a plate, petri dish, whatever, cells, take pictures every single cell, make a video out of it. In our body, it's much more complicated. Why? Because we don't only have 3 cells, we have many more types of cells. Right, let's talk about myeloid progenitor cells, the other arm of the hematopoietic stem cells. And what cells do we have? One, we can class them as either mononuclear cells, so the nucleus looks like one structure, or what we can say is polymorphic nuclear cell. So their nucleus looks many, rather than one single nucleus. The mononuclear cell we have is monocyte, okay, and they have phagocytic ability. Once they activated, they become as macrophages, okay, and their role, so monocytes once activated as macrophages, their role is to detect pathogen molecules, okay, to detect if their enemy is there, and also phagocytose them as well. So it has this phagocyte cytotoxic ability, and therefore it destructs them inside its cell, I'll show you in the next slide. And it can target literally pathogens that way. And then what happens after killing them, it acts as a signal for other cells, such as T cells, T helper cells. It'll bring some of those molecules of the foreign bacteria, pathogen, on its surface, and it acts as a radar, as a signal to other immune cells. Okay, and for instance, it interacts with the T helper cells to activate them. So here, you can see the mechanism. For instance, let's say we have a bacteria. This macrophage, what it does, it engulfs it into a phagosome like this. Okay, is this bacteria inside the cell or not? Is it? Sorry, wrong question. Is it inside the cell?
Yes.
Is it inside the cytoplasm?
No, it's inside the.
Well done. Question: why is it not inside the cytoplasm?
Because it is covered by the.
Yeah, it's in the phagosome. But why did this bacteria, this cell, did not take it straight into its cytoplasm?
Because maybe it can escape or infiltrate.
Well, because it doesn't know what exactly it does. Okay? So what it needs to do, because if it's inside, such as like let's say HIV virus, it needs to integrate inside the cytoplasm and uses our machinery to expand, colonize, and everything. And we don't want this to happen. So what does it? It brings it in containedly. Okay, it's a phagosome, brings all these lysosomes, kills it, puts it in tiny little pieces, and then puts it on these molecules called MHC molecules. We'll literally talk more about them tomorrow, but we have a 2-hour lecture on this as well, how this mechanism happens. It brings some of the molecules of this pathogen onto its these MHC molecules, and now brings it on its surface. Now what does this happen? This activates CD4 T helper cells. Okay, now this cell, having that antigen, having that molecule presented by this, now T cell receptor has a receptor for this and can get recognized. So, role of macrophage, what does it do? It kills the bacteria. It also acts as an antigen presenting cell towards T helper cells. Okay? And you can see here, this is a macrophage, and then having these dendrites like these. One question, why the macrophage has these dendrites? Why do you think?
So they can identify the other bacteria?
So they can have extra surface. Okay? So the cell might be here, but it also can recognize stuff over there. Because on these dendrites, what they're made of, they're literally plasma membrane. Yeah, they're the membrane of the cell. Yeah? But it has loads of receptors, and it has receptors here that recognizes the pathogen. So based on the amount of concentration of signal from the bacteria, you can see where it is. Similar to that cytokine from that route. Why? So we can reach it, grab it, bring it inside, engulf it, digest it, present it to other cells, and say, there's a foreign molecule hanging around here, looking like this. T helper cells, which one of you can recognize it? Can you recognize it? Yeah? And then initiates the whole immunity process. So this is macrophages. So now let's talk about the other cell. Before that, macrophages, we can find them in different tissues. For instance, we can have intestinal macrophages, alveolar macrophages are in the lungs, microglia is very important in the central nervous system, okay, and osteoclast. Macrophages, we'll have a lecture on this, but they can have 2 different roles. One is immunity, okay, fighting against the pathogen. One is immune regulation as well. So it has both roles. But you'll have a 2-hour lecture on this anyways. Right, the other cells, dendritic cells. Okay, so dendritic cells, if you remember hematopoietic stem cell, we have myeloid or lymphoid, and we can have dendritic cells from both. You remember lymphoid progenitors as well as myeloid. And based on the dendritic cells are in which tissue, they are named differently. Langerhans cells, lymphoid dendritic cells we can have. What are the roles? So DCs, if you think of that Ministry of Defense, I said, because many of my examples in my classes are military based. We're talking about immune system at the end of the day. So they are, I would say they're like radars. Okay? And I think it's in the next slide, you see a video. So their role is Radar, what does radar do? Send signal, hit something, comes back, realize who's where, okay? And usually radars are still in one location. These are very smart, they're moving around, okay? Yes, and you see. And once they get the signal, they become antigen presenting cells themselves. They go and activate the immune system. It's not just like, oh, there's a signal. They go and take the signal to the right location as well, okay, and give the right order. And one mature dendritic cell can activate around 100 to 3,000 T cells. One single dendritic cell can activate that number of T cells. And if you remember, I said CD4 T helper cells. One of them activating, it can activate many of them. So now you realize how efficient the whole system is and what Allah has created in us to have all these mechanisms. So in this video, this is going to be a live video inside the animal. Okay? So it's literally foot of a mouse. Here on the red, you can see the blood system. The blue is showing the collagen fibers. The green are dendritic cells. What has happened? They've made a cut here and put an infection. Okay? And look what happens to these dendritic cells. Or radars, I mentioned. All of them migrating here. Okay? And this is happening in vivo. So it's not in vitro in a plate. Yeah? Inside the foot of the mouse, everything trying to come here. And as you can see, they have dendrites as well. Why? Extra surface for extra attachment, extra communication, and also extra signaling to receive and send. Okay? So this is dendritic cells of today. Neutrophils. If you remember, they were the most abundant cells in the white blood cells, in the leukocytes. Yeah. First, they're polymorphic, so the nucleus is three-lobed shaped. It's not one single round nucleus. And they're one of the first cells that once the enemy attacks, these are the first cells that go and target, one of the first cells to target the pathogens. And on this video, what you can see, you can see a neutrophil, okay, red blood cells, white blood cells, and you can see this pathogen here, okay? So red blood cells, these are white blood cells, and this is, look at this trying to evade, this bacteria trying to run away, okay? And again, this is in vitro, it's not in a living human body. In human body, it happens much quicker. So it's just trying to chase it, the bacteria running away, okay? Look, it got trapped on this cell bit, went in there, then kill it. Okay? This is what neutrophil. Again, this is in vitro. In live body is much more efficient and optimized. Because in vitro is just literally imagine like a glass cell, whereas in the body we have all these proteins, matrices and everything helping each other. So this is neutrophils, okay? Eosinophils, they're again polymorphic, very, very huge cells. And they have a role in killing against parasites and also a very important role in asthma and allergy. Generally, in all conditions, they're found in thymus and spleen, ovaries. Once they become activated, then they're professional killers. So again, if I want to give an example in terms of military, neutrophils, eosinophils, or next slide, basophils, they're like tanks. Lymphocytes, they're like commandos, foot soldiers in a sense. They have destructive ability, fighting big things, okay? And whereas T helper cells, they are more signaling, sending signal, activating the rest of the immune system. Right, basophils, again, polymorphic, big cells, okay? And they have all these cells that I'm talking from neutrophils, eosinophils, basophils, they all have granulocytic activity as well. So inside them, they'll have all these granules. What is inside those granules? Loads of enzymes and killing products that kill these pathogens. They engulf it, kill it. Yeah? And the difference between basophils and the previous one, it also has histamine as well. And what does histamine do? Increases vascular permeability and causes smooth muscle contraction. For whether getting rid of the big pathogens, okay, whether it's through smooth muscle contraction is for excretion, okay, or sometimes we need it in vomiting as well. So, and also they secrete some cytokines which are not needed for today's session. Right, so these are the cells of the immune system, okay. What cells we talked? Remember? Lymphocytes first we talked about T lymphocytes. We said T lymphocytes can be T helper cells, cytotoxic, and we talked about B cells. Natural killer cells are regarded as lymphoid, but they're in the innate. Then we talked about dendritic cells. We talked about monocytes, activated, they become macrophages. And then there are mononuclear cells. Polymorphic nucleuses or polynuclear cells, we said what we have? Neutrophils was not the big one and they're the most abundant one. Then other than neutrophils, eosinophils, basophils. Yeah? Right, let's talk about organs of the immune system. So the basis of this Ministry of Defense of our body. So we have primary lymphoid organs and secondary lymphoid organs. What happens in primary lymphoid organ, which is bone marrow and thymus? This is the maturation site. In our primary lymphoid organs is where these cells become mature, whether it's bone marrow, whether it's in thymus. So in thymus, we have the T cells, yeah, bone marrow B cells, yeah. Hematopoietic stem cells, where they are? Bones, yes, in the bone marrow. But what happens, and do you remember some of them become T lymphoid progenitor cells? They migrate to the thymus, they become matured and active organs. Secondary lymphoid organs, they are like in spleen, lymph nodes, mucosal associated lymph tissue or gut associated lymph tissue. If you want to give a military example of that, so you know, like in sometimes in the big cities we have big military bases that all the military personnel go there for training and everything. So this is primary. Secondary are the ones which are more closer to the location of site that they need to show their action. And sometimes the enemy was not coming through here, but now they're trying to come. We go and create a new base. Okay, so that base was not there before. We established a new one. We also have this in our immune system. I personally did experiments where I did skin transplantation in the mice, and in the location that we're putting the skin is acting as what? A pathogen to the body, because it's foreign, it's not from that, it's a mouse. There is no lymph node, okay, but once we put it, because there is no close lymph nodes to it, sometimes new lymph nodes were generated. So with them we call tertiary lymph nodes. Secondary lymph nodes are like these, then these were tertiary lymph nodes. So this could happen as well. What is the role of these secondary lymphoid organs? It's the location. Firstly, we have a majority of these lymphocytes are residing there. It traps the antigen, okay, and then it presents it to the cells, the T cells or B cells, which one of you can recognize. What happens to T or B cells? They start proliferating if they can recognize it, colonize themselves to have more number and show their function. And then, so therefore, lymphocyte proliferation happens in these lymph nodes. So whenever you have flu or something, you go to the GP or to the doctor, what do they do? They touch here. What are they touching? Surgical lymph nodes. What they want to know? If they're swollen or not. If they're swollen, they know that, okay, you have been infected with some airway tract sort of viruses or something. Right, so in the thymus, which is a bilobed organ on the top of the heart and reaches maximum size during puberty, this is around 95 to 99%, the majority of it is T lymphocytes you have there that die in the thymus. Why T lymphocytes die inside the thymus? Anyone think why? What does it mean? So in the thymus, remember I said it's the maturation site, so T cells mature over there.
Yes.
But those T cells which have receptor against our own molecule, okay, they die over there. They get the signal to die or become inactive. Why? Because if they come to the circulatory system, they recognize ourselves and start attacking ourselves.
Is that when cancer happens?
That's when autoimmunity happens. Yeah? So this is what happens there. Okay, one of the things, how this happens, is called in the tolerance lecture. You have 2-hour lecture on that as well. They'll teach you what's the mechanism, how it happens, and if it happens wrong, what will happen. Okay, that we have patients with autoimmune diseases. And the structure, if you look at it, so it has cortex. So this region here is called cortex. This region in the middle called medulla. And you have literally, it's a network of cells gathering each other. Why they're sort of clustered in this way, in the lecture that you have on T cell development, you realize. They'll teach you about the mechanism and the microstructure of all this. So it's not it's too much detail for my lecture. So in the bone marrow, the whole process happens, okay? Maturation of these cells, except the T cell porogenesis that we mentioned, they go towards the other side. We have lymphatics. Lymphatics means the vessels connecting the systems. So for instance, let's give example of Palestine, a good example. So in Gaza, what's happening right now? We have roads. How Palestinians are fighting Israel? They have their VIP routes, which is tunnels. Okay. Immune system have how they can access to different locations. One is through circulatory system. Yeah. So we can have proteins, we can have lymphocytes, we can have cells inside the circulatory system to go to different locations. The other thing, they have their own channels of system called lymphatics. And usually how it works, so imagine this is a connective tissue around here. So we have lymphatics into the space in that connective tissue or muscle, whatever you have. And what can happen, cells can enter here, and also antigens and dendritic cells, for instance, can, let's say this is the skin, okay? We have the skin up here, and then there is a cut in here in the skin. The dendritic cells take a bit of that antigen, come here, migrate through these vessels, which they're called afferent lymphatic vessels, and what happens? They reach to a lymph node. Okay? Remember cervical lymph node, for instance? And why these cervical lymph nodes get activated? Because the virus comes through airway. The closest lymph node towards them for us is cervical lymph nodes. Yeah? And therefore they get swollen. So what happens? Again, dendritic cells brings the antigen through these afferent lymphatics, comes in here, here's full of B cells and T cells, and says, which one, we have a foreign molecule attacking us, which one if you can recognize it, which one of T cells? T cells, each of them come, and each of them have different receptor structure. One T cell receptor is this way, the other one is this way, the other one's like this. Why we have different types of T cell receptors?
To identify dangers.
Well done too, because it's a diverse response. Remember? We said adaptive is much, much more specific and diverse. And based on how that antigen looks like, we need to have the right receptor, so we need to have the right receptor on the T cell. If you can recognize it, this cell gets older to expand itself, so it has loads more of itself. Now these cells started showing their function. Activating B cells, activating cytotoxic lymphocytes, even recruiting more innate cells as well. So this is the lymphatic system that we have, and if you can see, the lymphatic system and the lymph nodes is all branched out in our body as well, whether human or even in animals as well. This is a cartoon of lymph node. You can see that it's made of cortex, paracortex, and medulla in the middle. And again, what happens in each of them in the B cell development lecture, they talk to you about it. But it's very, in basic terms, if I want to say, a lymph node has multiple entry points called afferent and one exit lymphatics called efferent. E for exit, this is how I remember it, exit, efferent. So one exit, multiple entry to the lymph node, and in the cortex we have B cells, dendritic cells, macrophages. Paracortex we have more T helper cells. Medulla we have plasma cells. What is the role of plasma cells? From school time, you remember?
They make antibodies.
Well done, they make antibody. Yeah? And what happens? They secrete these antibody, or even sometimes the immune cells, they either go to this, it has circulatory system connected to it as well, to the lymph node. So it can have access to the circulatory, as well as it can have access to the efferent, the exit lymphatic system. Okay? And that circulatory system is called post-capillary vein. Right, I think we covered everything on this slide. Spleen, the other sort of organ of immune system, is compared to lymph nodes is much more vascularized. Okay, and what does it do? It's literally traps antigen. It passes on the antigen, activating various different immune cells. And if you look at, you know, like the Kish airport or any other airport you have a control room on the top. Spleen's kind of acting like that. You can see everything because it has very strong and rich sort of blood circulatory system connected to it, as well as lymphatics. So literally can grab loads more sort of what do we call it, signals and molecules. So this is the spleen. It's highly connected to the lymph nodes as well as to the bloodstream. And the bloodstream, where does it go? It goes into the tissues as well as the lymphatics that we have. So all connection in the literally all connected in the lymph nodes as well as the spleen itself. And any aged T cells that we have, that they're aged, they're not active as much, they get sort of the signal that they become energetic, they switch down in a sense. Okay, because they're not showing that much of their function, they're not that much active, therefore they get the signal to be switched off, and this happens also in the spleen as well. Right, this is what we covered. So let's do a summary from what we talked. So first we did a group activity to realize the importance of the immune system. Then we talked about the mechanisms we have in place, and then we related it to the immune system. We said we have innate and adaptive. What are the components in the innate? Natural killer cells. One person at a time. What? Well done. Dendritic cells or DC. Yes, well done. Macrophages, which are activated version of what cell? Monocyte. Well done. What are the cells we have in the innate components? Basophils, eosinophils, neutrophils. Yes. What are the components we have?
Epithelial cells.
Well done. Epithelial cells, physical barriers, chemical barriers that we have. Yeah. And what were the features of innate? Innate. Faster response and same response every single time. It's the initial response that we have. Yes. Less diversity. Well done. Very less diversity, and it has no memory. Yes. Limited memory. Very. Yes, it says limited memory. Why? Because the feature on the bacteria is there. It's encoded, so it has a receptor for it. Straight away it recognizes them. It's not like if the bacteria is in different condition, different situation, different antigens, it will not reach the same response. Whereas adaptive, tomorrow you'll learn, is so responsive to the extent that a single, CH3 molecule is attached to this virus, it can detect it. Okay? A CH3, one carbon, 3 hydrogen. Even to that extent, adaptive is more specific, whereas for innate, no. Right, what else? So this is innate. Adaptive, what else? B lymphocytes. What do they do? B lymphocytes, what do they do?
We directly kill the pathogens, but T lymphocyte bind the antibodies.
B cells, yes, they differentiate into plasma cells to make antibody. Antibody tomorrow we'll talk about. B cells, one very important thing they do, they act as antigen presenting cell as well, as you mentioned. T lymphocytes, what do they do?
Lymphocyte, the helper one?
Helper, we have T helpers. What are the marker?
CD4.
4. And what do they do?
They have the cytolytic cell to kill the pathogens.
They activate
They actually kind of T helpers
They
activate the whole
activate
immune
the
system.
whole.
Cytotoxic, what do they do?
They
Let you
actually
know,
do the pathogens.
They target the cells which are infected with virus or tumor cells directly and they kill them.
Myelolysis, they get
And once we have few of them, yes, in adaptive we have memory cells, regulatory well done, switches down the immune system. What were CD things that we mentioned?
CD19.
CD19 for B.
What does it stand for? Cluster of?
Cluster
Cluster of
differentiation. And it's the
differentiation.
receptor on the membrane of the
Yes.
cells. Some cells express them, some cells don't express them. Some cells express multiple of them. For instance, CD3 is expressed by both CD4 and CD8. Yeah? But what makes them different? CD4 express CD4, CD8 express CD8. Right, then we talked about each of these cells, what do they do? Okay. We talked about lymphocytes right now, what do they do? Macrophages, what do they do?
Detection and they eat.
Detection, targeting, yeah, and acting as an antigen presenting cell. Neutrophils, what do they do? Well known first line of cells that they go and we have the most abundant of them in our blood system. And eosinophils, basophils, their role, do you remember?
They are professional killers.
Both of them professional killers, yes, well done. Polymorphic, yes. About their role in the innate, you have 2-hour lecture on the innate as well. So for me, just know the brief understanding of what is that, it's fine. And what else? We talked about organs.
Yes.
Primary organs such as bone marrow, thymus. What happens there? Maturation site.
Maturation site.
Secondary, spleen, mucosal associated lymph tissue, gut associated lymph tissue. Yeah. And all the lymph nodes that we have. And what happens in the secondary lymphoid organs? Antigen presentation, yes. So antigen gets trapped and antigen is presented to which cells? T and B cells. T and B cells start expanding, which tomorrow we'll talk a little bit more. But again, don't worry, 2-hour lecture on this as well. That's why I'm saying it's all linked. Okay. Then the lymphatics we mentioned, the structure of thymus we talked about, it's medulla and cortex. And likewise, same with the lymph node. And what happens in the lymph nodes? The difference is we have multiple entries of lymphatics called afferent, and one single exit which is called efferent, and it's vascularized as well. Okay, and also likewise similar thing. Spleen does this, but in a bigger scale. And that's it. Yeah, so this is regarding what you learned for the exam and all that. So read the reference book. So go on Google the reference book. I tell you the name of the authors. You can read any of these books. One is Abul Abbas. The other one is Cuba. Yes. Which one? What's the name? Abul Abbas. Yes. What's the name of the book?
Abul Abbas.
Salam
Molecular. Yeah, that's the one. So that's one of the book. Okay. I don't know if you're a person you have to read anything here by book. I generally read them, download them for free on gen.lib.rus.ec. And one is Kubai, which is Kuby, the other author, Abul Abbas. Any of these books will do. And Janeway also, depends which one you like. Which personally, in my slides, I use both Janeway and Kubai and Abul Abbas. Okay, so my diagrams is from all three. Or Clinical Immunology as well as another book written by Roit, R-O-I-O-T. So these are the reference books. So the topic I covered, if I just had to read for you from the book, takes you maybe 8 hours. But how many hours was my lecture? 2 hours. Okay, so this is an overview of the textbook. And why it's important to come to the class? So it will help you to catalyze, to get the information so you can, when you read the book, you understand it better. Otherwise, yeah, you can start reading the book, but because there was not a person there to explain you and linking things together, because the parts I mentioned, it's the initial chapters as well as some chapters at the end, because they're linked together. So come to the lectures, understand the lectures, but read from the textbook to understand. If you do this, on the exam you'll get 100%. Okay? If you don't do this, just read the exam, memorize the exam questions, don't come to the lectures. You might pass as well. But the feedback I got from the medical students themselves, you continue this way, you'll get the pass mark, but then you realize you don't know anything. Why? Because you didn't understand it. So my advice to you, come lecture, understand, read the textbook, deeper understanding, you can write down your own notes, it's there.
I mean, so which book do you actually want to use?
Any of these. Any of them is
Okay, they all have the same information?
same. Different diagrams. One of them, for instance, Abul Abbas is a little bit more cellular, molecular.
So which
Janeway
book-
Kuba is less.
Which one would be more simplified?
Depends on person. Different person likes different ones.
So it depends if someone likes molecular or clinical.
Clinical, we have that clinical immunology. But sometimes if you straight away, it's two different languages. Sometimes some people like it very clinical, they enjoy it more. Some people want to know exact detail how things work.
So which should we prefer for the
Up to
future?
you, it's both. At the end of the day,
Molecular is mostly studied.
molecular, what does it do? From molecular and cellular comes to clinical, the other one from clinical goes to cellular and molecular.
I think molecular to clinical is better.
Depends, that's your choice. Some people like to know the disease first,
Yeah.
then go to the molecular. But this way, there are too many diseases, so molecules might be hanging around different places. Whereas knowing the building blocks first, it's you can apply it quickly. So it depends on. Right, how many are in the class? 1, 2, 3, 6.